So it does manifest as
So I'm really glad to have Professor Jeff Abbott, who's coming to us from University of California in Irvine, where he's professor of pharmacology and vice chair of the Department of Pharmacology, right? By the way, this is where our chairman of biomedical engineering is coming from. He used to be the chair of biomedical engineering there. So Jeff received his Ph.D. from University of London in biochemistry, and then came to this country actually to join the laboratory of Steve Goldstein at Yale, supported by the Burroughs or Wellcome Trust, the Wellcome Trust Fellowship. There he started to work on the KCNE family of genes and proteins, doing clonings and studying structural and functional details of this family, and that continues till today. And so thank you for coming to share this with us.
Okay, thank you very much for the invitation, and it's been a fantastic day touring the university and speaking to everyone. You've got a fantastic setup here. It's very impressive. Very impressive place. All right, so I've got a ton of stuff to talk about, and it's all over the place, so I'm going to get started. So I'll talk. So I realized when we put the talk together, I've been doing this for 10 years. Just one. That's 10 years since we published the first knockout of the KCNEs that we were studying. So I realized, well, we've been doing this for. Okay. So most of you many of you probably know. I'm just going to talk about brief introduction. So KCNE proteins, they're a family of regulatory or beta or ancillary or accessory subunits, whatever you want to call them. They interact with ion channel pore-forming subunits to change multiple different aspects of the function, the cell biology, the pharmacology, and regulation by other proteins of the channels. So these things, as we were just discussing, they're not essential for the channel to function. If you express the alpha subunit in a cell, it will make current. It will open and current will pass. But these in vivo, in specific cases, the KCNE proteins are essential for the channel to be able to carry out its biological function. So a very quick just reminder about voltage-gated potassium channels. They have 6 transmembrane domains. The alpha subunits, a voltage sensor. They have a pore loop structure here. 4 of these come together to form the tetrameric channel that's functional, and then the KCNE subunits fit in somehow, maybe 2 KCNEs to 4 alphas, maybe 4 to 4, maybe variable. I'm not going to get into that one here. There's 5 KCNE genes in the human KCNE gene family. Single transmembrane segment. They alter almost every aspect of channel biology you can think of. They're promiscuous. Each KCNE can regulate multiple different types of ion channel, including other non-potassium channels. Each KB channel that we know about that's regulated by KCNEs can be regulated by multiple different KCNEs. Very complex system, right? So you can co-express an alpha with a KCNE in a cell type and get an effect. But in most cases, but what does that really mean for biology? Okay, so I'll give you an example of some of the huge effects one can see. So here's KCNQ1, which is an alpha subunit which I keep coming back to, and we've been talking a lot about it today. Many people here are studying this fascinating alpha subunit. So it passes outward current. If you have low potassium outside and you depolarize the cell, voltage sensors are activated, they open the pore, and then potassium ions diffuse out, and KCNQ1 this is a 3-second pulse, relatively slowly activating at room temperature. When you add KCNE1, it becomes way, way more slowly activating. The macroscopic currents are increased, the voltage dependence is right shifted, the activation, which is difficult to see here, disappears altogether here, or maybe it's not in activation at all. And so huge effect. And in the heart, these complexes are very important. Now, there's another example here of KCNE III interacts with KCNQ1 and produces a very different effect. So here you can probably see that now you're getting current below the line. So this is basically at physiological conditions, the reversal potential for potassium is around about minus 80 millivolts. So that's why we have 0 current at minus 80. Here is for 2 reasons. There's no driving force for net potassium flux, but also none of these channels are open. In this case now, you can actually see that if you go more negative to minus 80, you can see current, because there's now driving force, and it shows you these channels are constitutively active. So Casein III, we and other people have shown over the years that Casein III locks open the voltage sensor of the potassium channel, tricks it into thinking that the cell's depolarized, so basically you pass current at quite a wide range of potentials. If you go low enough in voltage, more negative enough, you will see closing. You can actually see a tiny hint of some deactivation here when we went from minus 80 to minus 120. You can see some closing here. But, so you can see how much the KCNEs, these little subunits, change the properties of the channels. Now, here's the 5 KCNEs aligned as a transmembrane domain. So they've got this extracellular N-terminus, intracellular C-terminus. They've all got PKC consensus site, and we and others have shown in some of the KCNEs this is definitely functional. And I've shown you here, I've highlighted disease mutations. So this is, one of the reasons we are interested in these, apart from the fun of studying these channels, is that they mutations in the KCNE subunits are associated with human diseases, such as AF, atrial fibrillation, Brugada syndrome, and long QT syndrome. So I've just placed it. This was fairly current around 2014. There's a lot of mutations in some of these. That are associated with human arrhythmias. In some cases, actual linkage, where statistically is very robust association. In other cases, there might be a sporadic mutation. Maybe a family member has this sporadic mutation. They have an arrhythmia, so you can question the disease association if it's supported by physiological evidence showing that the mutation changes the current. You can be a little bit more sure. But some of them are quite shaky associations, maybe. But still, there's good evidence that these proteins are very important for the function of one or more cardiac potassium channels by regulating them in vivo in human heart. So, there's 40 different voltage-gated potassium channel alpha subunits in the human genome. There's 5 KCNE beta subunit genes, and they can all interact with one another in various different ways in multiple cell types in the body and in multiple different mechanisms with lots of different outcomes. Then the voltage-gated potassium channel alpha subunits can form heteromeric channels. So you don't just get one alpha subunit in a tetramer, a homomeric channel. They can interact with one another in different ways. So you can imagine the combinatorial complexity of all these different subunits. So we can keep ad infinitum co-expressing them together in CHO cells or oocytes and seeing what they do, but it doesn't really answer the big question of what's actually happening in vivo. So the problem is, we want to identify what these do in the body, but they're promiscuous and they're expressed in so many different tissues. So the approach we've taken in my lab is to basically, we have worked with clinicians and geneticists in the past to identify disease mutations in KCNE proteins. We have studied, I couldn't get animal protocol approval to put a mouse on top of a xenopus, so this isn't actually a xenopus, this is a photograph I took from the internet. It's basically, this is showing that we use frog eggs and we use knockout mouse models. So with this combination of minimalist through to this animal model, through to real life data from patients, we can start to build up an idea of what these things do in the body. And this is just to remind me to tell you that most of the data I showed today is focused on the mouse, so it's all about mice today. Okay, so we have studied several different, almost all actually, the KCNEs. We don't study Casein I because that was, the knockout was done a long time before I started studying these, and we've focused on the other ones. So I'm going to take you through 4 of these different knockout mouse models, show you data that might not all be cardiac, but a lot of it relates to the heart. And the main thing I'm kind of trying to get across is I guess, 2 things. It's the amount of different ways in which KCNEs can regulate ion channels and other proteins and systems. The other thing is that we think that cardiac arrhythmias are much more complicated than just the heart. So, I always like to write a number of review articles where I mention there's about 25 different genes associated with sudden cardiac death or arrhythmogenesis. Almost all of them are expressed in multiple tissues, not just the heart. So, people sometimes criticize us and say, why don't, you need to do a tissue-specific knockout. Well, I agree that can give you a lot of information, but humans, unless they have a somatic mutation in a gene, they are global knockouts, or at least global mutations, right? They have a global mutation. So you have to think about what the disruption of that gene can be doing in multiple different tissues and how they can all interact. So we are not, once you open this can of worms, it gets very complicated. So we're not at the end of the story probably for any of these systems, but I just want to show you. So I'm going to get, in order of decreasing order of KCNE numbering, increasing order of complexity, I'm going to just go through these 4 stories. Okay, so KCNE 5. So this is a collaboration we've been doing with Nicole Schmidt, who is in the University of Copenhagen, and she actually generated a KCNE 5 knockout mouse, and then asked us, one of her students came over for a year to study it in my lab, basically. So we breed the mice, and then we study the cardiac function of these mice. So KCNE 5, it's one that's been studied, less, because you kind of start on the KCNE 1 and you go through 2 and 3 and 4. So there's a few mutations associated with things like AF and Brugada syndrome, actually. So, and it's known to regulate casein Q1 from oocyte experiments in the past, and it's known to regulate KB4 channels as well, possibly in the human heart. We wanted to see what happens in the mice, where we can take a real animal, we can generate knockouts and see what happens. So I mentioned in the title, I think, that this is a X-linked. So this is an X-linked gene, so that straight away gives you some kind of complexity. So if we look at expression of KCNE5 in human heart, we can see it's expressed pretty well in the atria and ventricle. In the mouse heart, it's actually lower expression in the ventricle and more highly expressed in the atria. But we study both systems. So we generally do a kind of workup on the mice to make sure there's nothing completely bizarre going on before we start. Very often there is something completely bizarre going on with the knockouts, but in this case, the only thing we could see was that there's a lower body weight. Consistently, they have a lower body weight. So this symbol here means that this is a male, so I'm going to just report a male mice for the KCNE5. So this means they have a knockout of the X chromosome, and of course the Y chromosome is 0 because they don't have there's an X-linked gene. So these are essentially, they have no KCNE5. So we don't know what the mechanism for this is yet, but this is potentially actually very interesting, but it's another story. So the heart size all relate relative to the body weight in this case, so there's nothing bizarre going on structurally with the heart, we don't think. So just very simple stuff. We just isolate the cardiac myocytes. We're looking at ventricular myocytes here actually, and we see straight away when we look at the male, I'll call them knockout just for simplicity, we see that the currents are bigger. The potassium currents, when we do a family of depolarizations, we see that a whole cell myocyte potassium currents are bigger in the KCNE5s. So what we do generally to figure out what's happening, and a lot of this is based on Gina Bond's work actually, we basically use pharmacology to figure out which of the currents is changing.
Jeff?
Yes.
Question? Since females have normally 2 copies, do they have smaller KD currents than males?
That's a good question, and we haven't done that yet. It gets complicated though. It gets complicated. Yeah. The currents are all different in females compared to males anyway. We find it's all different. But yeah, that's a very good question, and we typically look at sex differences. In this case, we just focused on the males to start with.
Okay.
You get mosaicism in the females as well with X linkage, so it gets very complicated. So the simplest thing was to start with the males. So we basically see that there's actually an increase in the knockouts, so that's unusual. In the past, we've always seen decreases, but when we knock out one of the KCNEs. But here we see an increase, and we see that the current's tetraethyl ammonium sensitive, and there's no 4-aminopyridine sensitivity at all. And what we find, just to cut a long story short, is that Kb2.1, which is a tetraethyl ammonium sensitive portion of the IK slow current in the mouse ventricles, is upregulated by knocking out KCNE5. And if you look down here, here's the Kb2.1 current, and this is basically now taking a system and moving it to a CHO cell or a COS cell, and here we see Kb2.1 is normally highly suppressed by KCNE5. So in our mouse, when we knock out KCNE5, we increase the Kb2.1 current. And in fact, it's some of the mutations I showed you, especially this one here, which is L65F. This is in the transmembrane domain. You can see that the human mutation, we get increased Kb2.1 current just like we get in our knockout. Now that mutation, the mutation of KCNE5 are associated with Brugada and AF. So atrial fibrillation. So normally when we think about long QT syndrome, which is a ventricular arrhythmia where repolarization is delayed, we think about loss of function of potassium channels. So or gain of function of late sodium current. So if the potassium channel is not working very well, they're the ones that normally repolarize the myocardium. So if you reduce their function with an inherited mutation or drug block, you extend the repolarization period. Well, atrial fibrillation in Brugada to some extent the opposite of this. So actually a shortened effective refractory period predisposes to atrial fibrillation, and in Brugada syndrome is very often loss of function of sodium current or gain of function of potassium current. So in our mouse model, by knocking out Casein I, we're actually increasing potassium current, which would be more consistent with what we see in human patients, such as L65, where we see an increase in potassium current. So that's what I'm going to show about that. We're in the process of revisions on this paper. We have a ton of biochemistry and cell biology data, which makes this story a little bit more interesting. I don't have time to go over it today, but just want to show you that's one of the examples where we think that it's a myocyte defect, and it's fairly straightforward. All right, so I'm going to move on to Casein IV. So Casein IV is actually so far relatively little disease association in humans. So there's an atrial fibrillation has been associated with one point mutant here. It's a variant, and in several different Chinese studies, it's been shown that this predisposes, depending on what residue you have here, predisposes to atrial fibrillation in large populations. But there's never been a real genetic linkage study done. So we wanted to knock out Casein IV and see what happens. So the first thing, so we actually collaborate with Steve Goldstein, my former mentor on this. He and Dan Levy had been studying this. They'd made the mouse and then they kind of moved on to other stuff, and so we gladly took the mouse and started studying it. And we've been studying this mouse for a long time because it's quite complicated. So after our initial studies of knockout mice, we realized that we were being very naive about the way we were studying them. And we realized that you have to look at the age and you have to look at the sex of the mice. It's really important. And the KCNEs are actually highly regulated by a number of things, including hormones. And so it makes a massive difference. And we really learned, one of the big lessons has been you have to be extremely careful, not just about the time of day that you're studying them because of circadian rhythms, but you have to also look at the age and the sex of the mice, and you get completely different results if you don't do that. So one of the first things we saw, just making sure that a gene was actually expressed in the heart. You can see that in the females, there's hardly anything there. So these are sexually mature young female. There's hardly anything there, whereas the males have high expression, especially in the ventricles. So this is a major difference which you just wouldn't see if you weren't being careful about the sex of the mice. So we looked at both male and female currents, and we can see that this is again isolating ventricular myocytes from wild type and knockout. This is young, sexually mature male and females. We can see, as one might expect, so it's very nice. In the females, there's no difference in the current, which you might expect because you've knocked out subunit and it wasn't expressed at the ventricle to start with. In the males, there is a difference, and this time it drops. So you get a drop in current density. And actually, interestingly, it brings it down, brings the male currents, and this points to Colin's question, this brings down the current density in the male parts to that of the female. So in several different species, the male ventricular currents are known to be larger than the female, actually. And in this case, we can see that Casein IV might actually make the difference because if you knock it out, it drops them down. And there's no real, didn't seem to be much of a difference in the steady, sort of steady state-ish currents. So we did some pharmacology and some curve fitting to figure out which currents were being altered. So we see if we fit the currents, the ventricular currents. With the triple exponential from Gina Bond's work, we can see that we have the ITOF is reduced by about 50%. So this is the fast transient outward current that gives you, in mice, everything, obviously they have a much higher heart rate than humans, about 5 to 10 times faster. So there's no plateau phase, it's very, very, very rapid, and much of it's dependent on ITO, actually. So we see a 50% drop in that current. Then we also see the drop maybe 40% in IK slow as well. So in the mouse heart, the Kv4, just like in the human heart, is Kv4.2 and or 4.3 generates the ITOF. And KCNE4 was already known to regulate Kv4 channels, and is speculated to do so in vivo in humans as well, but it certainly does in vitro. So that was very consistent with what was already known. But we were interested in the IK slow because that was actually not previously been looked at. So again, we went back to look at 4-aminopyridine and tetraethyl ammonium. And remember I told you that in the KCNE5, when you knock it out, you increase the tetraethyl ammonium sensitive currents. Well, we didn't see any effect on the TEA sensitive currents. In fact, we saw a reduction in the 4-aminopyridine sensitive, which are Kv1.5. So it looks like, from our data, and we back this up by co-expressing the subunits together in CHO cells afterwards, and the results make sense, that basically KCNE4 in vivo, in the mouse heart at least, regulates Kv4 channels and also Kv1.5, which was actually not known before. Now, remember I told you that in humans, it seems like KCNE4 was also important in atria. So we looked at atriomyocytes as well, which is a lot more tricky in mice because the atria are quite small, you don't get quite as many cells, and they're not usually as robust as a ventricular myocyte. But we did this, and we saw a reduction actually, this time not really much happening in IK slow, but we saw a reduction, a 50% reduction in ITOF again. So we think that in the atria, maybe it's a slightly different story than the ventricular myocytes, but there's still a reduction. Now, this would not be predicted to cause atrial fibrillation because we're getting a decrease in the current. So we don't think this could ever explain what's happening in human beings, but we think mice are very different from humans, and in human beings there's a lot of other currents such as IKS, which is KCNQ1 plus KCNE1, and also the hERG potassium channel, which really don't really get expressed much, if at all, in mouse, ventricle, or atria. So it's very possible that the effects of KCNE4 on KCNQ1 are more important in human beings in terms of mediating any association with atrial fibrillation. But we like using the mice because we can. It's a system that one can experimentally manipulate, and we see very clear effects on the ITO current in both atria and ventricles, and this novel association with Kv1.5. So, okay, so that all made sense. So it takes young, sexually mature male and female mice. The expression in the females is very low. We don't see any effects in the females at the cellular level, and in the males we see expression of KCNE4, and we see quite robust effects in the currents. So we went to do electrocardiograms, and because we had quite a lot of mice piled up by this stage, we thought we'd look at several different ages. So we looked at the 4-month-old, and we looked at 12-months-old, and we looked at 17-months-old. And what you might notice here, first of all, we always see that in mice, the QT interval gets longer and longer as they get older, even in wild mice. So they become kind of more susceptible to long QT syndrome as they get older. But the other thing we noticed was that in the female mice, there's now a QT prolongation in the knockouts. There's a QT prolongation in the knockouts just in the older mice. So there's no real effects in the 4-month-old or the 12-month-old, but 17 months is actually QT prolongation. So KCNE4 suddenly becomes important. So what's different for about 17 months in mice compared to 12 months? Well, that's actually postmenopausal in female mice. 17 months is postmenopausal. So postmenopausal in mice, dihydrotestosterone levels actually go up. So then we have the idea that, okay, maybe this is being regulated, KCNE4 is being regulated by DHT. So another group, Mansouri Iqbali, a long time ago, showed that KCNE2 is highly upregulated by estrogen, actually, in the heart, which is very interesting. We haven't really studied it that much. But we stumbled upon this and kind of think maybe this is the opposite. The yin and the yang is maybe the KCNE4 is the male version of KCNE2. So what we did, we took mice, these poor mice, male mice, castrated them, and then in half of them we gave them DHT pellets, so as a control. So now you've got some mice that are castrated very early on, so males, so they don't have high levels of DHT, and then you have some where you do the castration as a control and you put back DHT pellets, so they have normal or higher than normal levels of DHT. And then we looked at real-time PCR to measure the KCNE4 levels in the heart. Sure enough, I'll just run you through this. At the The young, sexually mature male and females, we see nice expression of KCNE4 in the wild type. You use the knockout as a control, obviously, so there's no expression there, so we know we're looking at the right gene. Females, there's hardly any expression, as I showed you before. Then if we look at males 17 months old, so the DHT levels drop a bit in these older male mice, but not dramatically. So nothing like the DHT levels in the younger females. So they still have robust KCNE4 expression. The postmenopausal female mice, they now have high levels of KCNE4, just like the younger sexually mature male mice and the older mice. So that was one way of doing it. Then we also looked at the DHT, so the castrated male mice without DHT pellet, they have low levels, quite similar to the younger females. And then as we leave the pelleted mice, as we leave them longer, we see the KCNE4. This is a slow release of DHT. The KCNE4 levels come back. So we think that the KCNE4 is actually a DHT regulated KCNE subunit, and it's kind of very interesting for us because we think there might be some reason why males and females need to regulate their ion channels differently and at different stages. So they can upregulate KCNE2 via estrogen, and upregulate KCNE4 via DHT. And just to bring this back to human populations, one of the things that came out of the Framingham trial was that so the particularly sensitive, or particularly the population in the Framingham trial of older men that were particularly susceptible to AF were those ones with low testosterone. So this could be part of the mechanism. So just to speculate, KCNE4 reduction is associated with this atrial fibrillation in people. And so perhaps the older men that have low testosterone also have low KCNE4, and then that much more susceptible. So we know that in aging, your susceptibility to AF goes up, and it's a disease which is getting more and more prevalent in the aging population, as we talked about. We think that maybe testosterone level could be part of the picture, at least in men. So it might be something to examine. So after we published this work,
can you give
yes.
some DHT to females? To
We
young
haven't done
females?
that. We have not done that. And there's other experiments we could do as well. We could over-rectimize the females as well. So we figured that the shortest route to get to this answer was to have if you have the castrated mice and then the castrated with DHT pellet as a control, it's a pretty good control. But yeah, sure, there's other experiments we could do to follow that up even more. Absolutely. So after we published this study, we were contacted by Thomas Jeps, who's a guy who works at the University of Copenhagen, and he just found out that KCNE4 is very important in regulating KCNQ4 in rat arteries. So when they look at, one of the things they look at is the mesenteric artery. So the KCNQ1 that we know about is highly expressed in the ventricles, is important in ventricular repolarization, atrial fibrillation as well. But the rest of the KCNQ family, historically, we thought of as being neuronal channels, and more and more it's emerging. They're actually very important blood vessels, the KCNQs, maybe 2, 3, 4, and 5, depending on which paper you read. So Thomas Jeps has been studying this in the University of Copenhagen, and they use a technique where they already knew that KCNQs were involved in the arteries, and then they started looking at KCNEs, and they found that if they use morpholinos to downregulate KCNE4, they change the function of the KCNQ4 channel, and KCNQ4 is important for the relaxation of the arterial smooth muscle cells. So when you don't have KCNE4, these channels actually don't get to the membrane very well. You tend to move towards contraction of the smooth muscle. So when he found out we had a KCNE4 knockout, he said, "Can we knocked him out with morpholinos, but how about looking at your mice where you have a global knockout?" So he came over, and within a few weeks he got beautiful data. So when we split them up into male and female, obviously, and so he uses myography. So he's basically looking at changes in where he holds small sections of the mesenteric artery, and then he's looking at the force generated here in the millinewtons. And what you can see here, so these are basically wild type in black and KCNE4 knockouts in red, and he gives methoxamine, which is an alpha-1 adrenergic receptor agonist. And what you can see is that here's the wild type contracted. The knockouts are way more sensitive, so they're way easier to contract than the wild types. That's in the males. In the females, there's no difference. So you see over here, there's no difference. So we're very excited about this because straight away he recapitulated what we found in the mouse heart in a completely different system. So it looks like the same sensitivity is a difference in sensitivity, or difference in requirement of Casein-4 in males and females is also in the blood vessels. And so we saw a shift here in the dose response, and here's the average data showing the dose response, showing that the males are basically much more susceptible. So the other thing he did, he used an activator of KCNQ2 and Q4 channels, ML213. And again, you see a big shift. So they're much less sensitive to the activator because we think there's less Q4 channels at the membrane because E4 helps to traffic Q4 to the membrane. So in the females, again, there was no difference, and you can see the average data down here. So that's all. We're going to write this paper up soon because we haven't looked at KCNE4 expression yet. It's very nice, it's very consistent with what we found in mouse heart and what he found in rat arteries, but now the question is what's causing the male-female difference? Obviously the obvious answer is that it's KCNE4 is more highly expressed in the male arteries than the female, but we're going to follow this up with some real-time PCR and other work to see what's happening. But he's very excited about this because testosterone, there's a big sex-specific differences in vascular reactivity and in exercise, so you can improve vascular function and vascular tone by exercise. And you know, one of the things that intense exercise does, apart from improving vascular tone, is it stimulates testosterone release in males. So Tom's very excited about the possibility that KCNE4 could be part of the puzzle for why intense exercise actually increases vascular function so well. I think testosterone might be playing a role. And also, we all know males are much more susceptible to many of these vascular diseases. So again, they require KCNE4. All right. Okay, I'm going to try to speed up. So KCNE3. So I've told you about two of the KCNEs that are definitely expressed in ventricular myocytes and atrial myocytes, and what happens when you knock them out. So KCNE3. Again, there's association with Brugada syndrome, association with atrial fibrillation for KCNE3. When we knock KCNE3 out, we can see eventually, as they get older, we can see QT prolongation, especially in the females. So they do get some mild QT prolongation. Okay, we're going to figure out, we're going to follow this up, look at the mechanism. So we're very surprised when we isolated the myocytes and started studying them, we basically saw no difference. There's a small difference in the tau of an activation up there, but if you study enough things, you're going to see a difference in something. So we don't necessarily believe anything's happening there. We saw no difference in ventricular myocyte currents at all. We did the drugs, we did everything, we see no difference. There's a really good reason for that, is because we see no expression of KCNE3 in the heart at all. So it's highly expressed in the colon. KCNE3 is highly expressed in the colon, but in the heart of male and female, it's not there at all. But we do see QT prolongation in the knockouts. So when we take the myocytes out of the body, we see no differences at all. But in the whole animal, we see QT prolongation once they get to about 9 months old. So what's happening? So we started studying many more aspects of these mice to figure out what the mechanism could be. And we saw, okay, sometimes we found the KCNE2 knockouts, that angiotensin 2 is increased, we see no difference in angiotensin 2. We looked at aldosterone, which we've also examined before in mice to look at potential changes in potassium in the serum. And we do see a doubling in aldosterone, which suggested that the serum potassium might be different, and this is in response to that. But when we looked at serum potassium, there's no difference. In the male or females, knocking out Casein III does nothing. We also saw some kind of dyslipidemia, and we're not sure whether that's part of this mechanism I'm talking about, but it's just interesting. We see basically high LDL. This is really the only thing we had to go on, that the mice have double the level of aldosterone without a change in serum potassium. We looked at potential innervation differences, nothing, glucose is normal, there's no glucose intolerance. So we looked at the adrenal glands, which secrete aldosterone, and there's no, Casein III is not expressed in the adrenal glands. So we're very confused. So in my lab, one of the techniques we've been using, because it helps kind of in detective work when you're figuring out what's going on with the mice. You basically take the wild type and the knockout tissue, and you do microarray analysis and look at expression of all 28,000 genes to see if there's any patterns emerge. So you basically compare the complete genome transcriptome expression in the wild type and knockout. And we did a few adrenal glands, because of the aldosterone thing. We did a few adrenal glands from the wild type and knockout. And what we found was that a very small subset of genes were changed. Transcript expression was changed between the wild type and knockout, but none of them were really genes that one associates with adrenal gland function. In fact, they're all kind of immune cells or chemokines that attract immune cells. So things like STAT4, for instance. There's actually a gene up there which might be interesting. So it actually looked as if when we looked up all the different functions, the genes that were changed, it looked like the adrenal glands were releasing some kind of chemokine, and that the genes that were upregulated in the adrenal gland were actually more reminiscent of activated B cell transcripts. So sure enough, and it doesn't come out very well here, but when we looked at the histology, we found that there was indeed an activated B cell invasion of the knockout adrenal glands. So we don't know, it's basically an autoimmune disease caused by Casein III knockout. And this seems completely bizarre. Now, at the same time we were studying this knockout mouse, my former postdoc Torsten in Berlin, he was studying the Casein III knockout mouse that was made completely independently. It was given to him by Thomas Jentsch. And, Torsten was studying atrial function. And I told him about our results, and he said, we think we see expression of Casein III in the heart. He went back and analyzed all the data again. He said, okay, you're right. There's no Casein III expression in the heart. I said, okay, look at the aldosterone and look at the adrenal glands. And he looked at the aldosterone levels. He found they're raised in this other colony, just like ours. And he looks at the adrenal glands, and they also have an activated B cell infiltration. So in two completely independent mouse models in separate countries. So then we looked at spironolactone. So to block the effects of aldosterone, you can add spironolactone. We found that the QT prolongation went away. There's no differences in the EKG. And when we did a coronary artery ligation to kind of increase the arrhythmogenic phenotype, thank you very much, to increase the arrhythmogenic phenotype, that's great. We saw that untreated mice, knockouts, develop sustained ventricular tachycardia when we give them a ischemic insult, whereas the wild types are fine. If you treat them with spironolactone, you now get much shorter non-sustained ventricular tachycardias, and the statistical analysis shows you that the spironolactone is highly effective at preventing the arrhythmias. So this is bizarre. Now in human beings, Casein III is properly expressed in the heart, so it's different between mice and humans. Based on other people's data, it's fairly convincing to me that Casein III is expressed in the heart, and that the Brugada and possibly AF that's associated with Casein III is at least in some part due to a defect in ventricular myocytes. But at the same time, I don't think anyone's ever looked at those patients to look at their aldosterone levels or to look at potential activated B cell infiltration in the adrenal glands. So we think this is a case. It's nice to have the mouse model where Casein III is completely absent from the mouse heart, because it just shows you what can happen when you knock out a gene that's expressed in different parts of the body. So the classical, you disrupt your channel gene, you alter ventricular ion channel function, this change in ring polarization can cause abnormal rhythms, EADs, etc. Ventricular tachycardia can provide electrical substrate for sudden cardiac death. In the case of our mice, the Casein IIIs, we think the E3 disruption is actually causing some kind of adrenal autoimmune reaction, and now it's been backed up by the work from Torsten in a completely separate colony. We think this is causing hyperaldosteronism, impairing ventricular myocyte K channels by a completely different mechanism, providing electrical substrate for sudden cardiac death. And then also remember I mentioned they have hyperlipidemia as well. So this in the long run can cause myocardial ischemia and provide an ischemic substrate as well. So we started to think about this interaction between different organ systems. The Casein IIIs were just lucky because Casein III wasn't in the heart at all. So we could say this is definitely not a primary defect. Yes?
Have you looked at potassium levels though? Because spironolactone also elevates that.
Well, we didn't look after spironolactone treatment. We looked before. There's no difference. So there's no difference, and we don't know whether so Casein III is expressed in lymphocytes and other white blood cell types. So we think what's happening is it's causing a defect in the blood cells that may be telling them to attack the adrenal gland. Or we have no idea whether the attack of the autoimmune attack of the adrenal gland is in response to elevated aldosterone because of some other reason, or whether the elevated aldosterone occurs because of the autoimmune attack on the adrenal gland. We don't know. But whichever way, we think that the high aldosterone levels are basically required for the arrhythmogenesis. But yeah, we haven't looked at potassium after spironolactone.
Yeah, because it will increase potassium levels in the blood.
So you think it could be indirectly? Can be indirectly.
It could be secondary, not involved.
Right. That's very interesting. That's very interesting. Yeah, we haven't done it. All right. So in the time we have left, and I may not get through all this, I just want to tell you about Casein II. So this is the one we've been studying the longest. We've been studying for 10 years. And I think it's a very, very interesting mouse. There's mutations in Casein II in humans associated with long QT, also atrial fibrillation, and not so far Ruggiero syndrome. So Casein II, originally we found that it regulates hERG, and its collaborators found that Casein II mutations are associated with ventricular arrhythmias. Casein Q1 is another channel which most of us know because of it's regulated by Casein I in the heart, and that's important for the IKS current, and that's also KVLT1, Casein Q1, highly associated with long QT syndrome and also other arrhythmias as well. But the interesting thing is that Casein Q1 and E2 also get together to produce this very interesting channel. So Casein Q1 is a delayed rectifier. As I mentioned before, negative currents, hyperpolarized currents, membrane potentials, there's no current, and then as more channels open, you see more and more current. Q1, E2 makes the currents in Casein Q1 very small, but you see there's a linear IV curve. So it makes the channel constitutively active. So it still passes through the reversal potential for potassium, but now you can pass inward current, much like the Casein Q1, Casein III currents I told you about. So when we first knocked out Casein II, other people had shown that Casein II is expressed in the stomach, and they thought it was important for gastric acid secretion, and our knockout results completely validated all that work. So long story short, Casein II forms a potassium channel with Casein Q1 in parietal cells on the apical side. These secrete gastric acid. The HKTPase that secretes protons into the stomach lumen needs a pathway for the potassium ions that come through it to exit the cell again, otherwise the system breaks down. If you knock out Casein II or Casein Q1, you lose that recycling pathway and you completely lose gastric acid secretion. So here we show parietal cells that are loaded, proton loaded, and then allowed to secrete those protons with histamine and secretagogue, and they can recover their pH nicely. The knockouts, zero recovery. The parietal cell morphology also gets messed up by knocking out E2, but if we have heterozygous mice, we have normal morphology and we still lose gastric acid secretion, so we know it's not just a developmental or structural defect. So this potassium current is required, and it's a very striking effect, and the pH is about 6 in these mice of the stomach. So we found that if you keep studying these mice, we find that they actually develop this gastritis cystica profunda, which is they get huge cysts in the stomach, and they actually get gastric cancer, gastric neoplasia. So we published this in PLOS One about 5-6 years ago now. They get this adenomas, and then you actually see basically a neoplastic invasion of stomach epithelium into the blood vessels. So they get these invasions, and basically this causes gastric cancer in the mice. We had a lot of trouble getting this published. None of the cancer journals wanted to know. And then more recently, a Japanese group recently found gastric adenocarcinomas in people arising in gastritis cystica profunda patients present with selective loss of Casein II expression. So they look at the cysts, and the cysts just lose Casein II expression. So it looks like Casein II is definitely involved. There was also a very interesting paper in 2013 showing that Casein Q1, people with Casein Q1 mutations, we all know they get cardiac arrhythmias and get Jabel-Lang-Nielsen syndrome if they have a particularly bad effective mutation, which really reduces the currents. These people also get iron deficiency anemia, they get gastric hyperplasia, and they get elevated gastrin levels as well. So the same things that happen in the mice happen in people. And then very recently, Oncotarget, which is this thing which identifies drug targets for cancer, this journal, an individualized prognostic signature for gastric cancer patients for chemotherapy. They're looking at markers to see if they can identify which are the worst prognosis for patients. And it might be difficult to see here. They found 2 gene pairs. Basically, Casein II is in both the gene pairs. So if you look at, you can tell whether a patient is going to have a poor prognosis for gastric cancer solely by looking at Casein II and comparing it to different other marker genes. So I just wanted to show this, that the mice can be excellent predictors of what's going to happen in human populations. So when we knocked out Casein II, we saw a lot of bizarre defects, which I'm going to, because of time, I'm going to have to run through like a laundry list. But basically, if one of the original papers on Casein Q1 expression, we all think about it in the heart and then the stomach, the stomach rolls. If you look at the original, I think it was a PNAS paper where they did all human tissues and looked at Casein Q1 expression. The 2 tissues where it's most highly expressed are the stomach and the thyroid gland, and no one had ever studied it. So, and we hadn't either. We hadn't even thought about it. It's kind of hidden in this PNAS paper a long time ago. So we started finding that pups, our mouse pups, were bald. The knockouts were bald, especially if the mother was a knockout. So Torsten Ropke, the postdoc, was working on it in my lab. He got very impatient with studying het crosses, and he decided, I'm going to speed this process up. I'm going to start looking at knockout crosses. And it's not, you're not supposed to do that, but it led to this really nice discovery that basically Casein II deletion causes hypothyroidism. So here's a mouse who was completely bald, and because of hypothyroidism, and if you give it T3, T4 injections every other day for a couple of weeks, the hair grows back. You can see the follicles grow back, and you can also achieve the same thing by surrogating the mouse. So if you take a knockout pup and give it to a wild-type mother on day 1, so it can feed from the wild-type mother, it gets normal milk with thyroid hormone and iodine in, develops normally. So the mother's genotype was really important. So is kind of a nice thing to tell trainees is that, you should try something different. Don't always do exactly what you're told to do, as long as you keep within the protocol, safety protocol. This is the way you make discoveries, by doing something that you weren't supposed to, or looking for something unexpected. That's the way big discoveries are found. So we did iodide 124 accumulation kinetics using positron emission tomography in the dams, and we found, I don't think this is, no, it's okay. So what we found was, here's a positron emission tomography still from a mouse, that's the thyroid, that's the stomach, and that's the mammary glands. This is a lactating dam. So what we did, we used to inject iodine 124 into the dams, then we basically measure iodide uptake kinetics in the thyroid glands, and then we'd actually let the pups feed from them, and we'd do PET imaging of the pups as well. And we figured out the whole pathway doing this kind of getting more bang for the buck by injecting the mother, looking at milk uptake and iodide and everything in their mums, and then also looking at the pups. We found that iodide accumulation in the knockouts is way lower. So Q1 and E2 actually are required for iodide uptake in the thyroid, and we think that it sits here on the basolateral membrane, and it's important for function of the sodium iodide synculture in the thyroid gland. And this is the first role established for the cation channel in the thyroid gland. What I didn't tell you is that this affects cardiac function, no end as well. So Casein Q, and then Florian Lang's group about a year later came out with similar results using the Casein Q1 knockout. So it kind of proved the other half of the story. But this, as you can imagine, this thyroid defect has huge effects on the heart, and no one's really considering that. In humans, the thyroid hormone levels in people with Q1 mutations could actually be affected and could be causing indirectly effects on the heart. So the Casein II knockouts are, as I said, they're a very fascinating animal. So we started studying other aspects of the biology, and then a few papers started to filter through in the literature. There was a genome-wide association study in Nature Genetics showing that Casein II mutations are associated with early onset or a SNP, a polymorphism associated with early onset myocardial infarction in people. And then another paper came out, PLOS1, coronary artery disease is also linked to Casein II polymorphisms. And there's one down here as well, Casein II, this is a SNP very close to Casein II, associated with prevalence of myocardial infarction and long-term mortality. And there's been another study more recently in Saudis, in Arabs in Saudi Arabia, showing similar things. So we thought, well, we've got to look at the Casein II knockouts. So mice are very difficult to give atherosclerosis. They're very difficult. They're very resistant actually. But we could see even without opening up the vessels, just looking at the vessels, we could see things that look like plaques in the knockouts. So we were told that you shouldn't even bother studying this phenotype in mice because there's basically, if you look at single gene knockouts, there's only ApoE and LDL receptor knockouts cause coronary artery disease in mice. All the rest are kind of combinations and different diets. So there's really only 2 genes known in mice. There's a bunch more in humans. But when we looked, we had an undergraduate who had no idea what she was looking for. Brilliant blinded study. We just told her, just quantify this stuff, and her data is very convincing. Basically, the Casein II knockout predisposes highly to atherosclerotic plaques. These are Sudan-4 stained coronary arteries opened up on fast, and then we basically look at the staining, and we can see, I think very convincingly, that Casein II knockout increases the plaque surface area, and then if you give them a Western diet as well, it really increases it even more, and very much so in the females. So females typically are thought of being as less susceptible, but you remember I told you that Casein II is upregulated by estrogen. So we think that just like the males require Casein IV, we think that the females require Casein II. This is just looking at the contractile properties of knockouts. And knockouts are much worse at relaxing the vessels than the wild types. It's just a close-up so you can see the Casein II knockout really developed plaques. We published this last year in JMCC. So I'm going to have to stop soon, so I'm going to have to skip a lot of data. But one of the other things we've been studying, so we think that the Casein II knockouts have a multisystem syndrome that predisposes to sudden cardiac death from a variety of different angles. So they do have a primary defect in ventricular repolarization. They also have hyperlipidemia, and they get atherosclerosis. So they also become diabetic. So this is data. They're not quite diabetic yet in this data by the kind of accepted norm for mice, but when we leave them another few weeks, they're diabetic. In this data, we show you basically as soon as they were weaned, half the mice were put on a Western diet, which is why I have that on the poster. And the rest of the mice were on a normal diet, and you can see the wild types tolerate the Western diet fine. This is only 2 weeks after being on a Western diet. Knockouts, they're already starting to get glucose intolerant. And this is 21 weeks, and you can see they're highly glucose intolerant, the knockouts. So we think actually, I don't have time to show you the data today, they have a primary insulin secretion defect. We're measuring insulin secretion in islet cells, and Casein II knockout causes a primary defect in insulin secretion. And we actually think that Casein II regulates Casein Q1 in islet cells. So that's something we're currently studying. We don't have all the mechanisms yet.
Excuse me?
Yes?
Is the gene expressed in pancreatic beta cells?
Yes, it's expressed in beta cells.
So when you knock out, you expected that there is less insulin secretion.
We didn't really know. We didn't really know. We saw. I mean, it's kind of people have been trying to figure out what Casein Q1 is doing in the pancreas for a long time, and the results didn't always make sense. But we think it's because Casein II is regulating it. So it's kind of makes the current very different, basically. So we didn't expect that at all. So after finding glucose intolerance, we then went back and looked at islet cells, and we found first we went back and looked at whole pancreas, and we found that E2 is there. Now we followed that up by learning how to isolate islet cells, and we found it's in islet cells as well. So yeah, now you would expect it.
And the insulin secretion that you did was in isolated islet, or the insulin that you measured was plasma insulin? Because it could be expressed in the brain.
This is isolated islet cells.
Isolated.
Yeah, this is isolated. So we patch clamp them as well, and we see a huge change in the currents when in the knockouts compared to the wild types, and we also do isolate insulin secretion in response to glucose, and now we're going to do a response. I think this week they're doing IK, and also we're going to look at Chrominol 293B, which is a selective Casein Q1, fairly selective Casein Q1 blocker, to see if we're right that it's regulating Casein Q1, then we should see a change in the way it responds to Chrominol. So we're doing that. So I have way too much stuff, and I went really slowly over it. So I'm not going to show you any of this, unfortunately. I will show you this because it really relates to. Yeah, okay. Share this. So the other thing that happens with Casein II knockouts is that after 3 weeks they already have fatty liver. And long story short for this one is that we think we figured out the pathway. So because I mentioned the KCNQ1 mutations or polymorphisms in people can cause anemia. So we think what happens is in the KCNQ2 knockouts, they have no gastric acid secretion. That means they can't absorb iron properly, naturally. And they don't absorb vitamin B12 either, so that's why they get hyperhomocystinemia, which is another risk factor for coronary artery disease. But they get anemic because they have no gastric acid secretion. Now, we think that the anemia is actually causing them to have fatty liver, because iron's responsible, is important for a lot of the enzymes that are important in lipid metabolism in the liver. So we actually, using oil red O staining and a lot of other techniques which I've skipped through, we found that the KCNQ2 knockouts have fatty liver. There's the wild type's normal. If we treat them with injectable iron, which is very difficult to dose in small pups, but we managed it after a while, we see no change, we see no difference between wild type and knockout. So we think that the anemia is causing them to have fatty liver, and we think that contributes to the coronary artery disease, along with the insulin secretion problems that they have, and probably multiple other problems as well. So the reason I had a burger on the poster was because, in mouse models it's very difficult to get ventricular arrhythmias without kind of really taking them to the extremes. But what we found, the student Sumin Lee, who was working on this, he came to me one day and said, he said, the traces have all this noise. He was looking at the average of hundreds and hundreds of seconds or whatever traces in the EKGs. He said, I can't use this data. It's really noisy. It's always blurring. I said, well, let's have a look at it. And what it was actually, the noise he was actually looking at was in the female knockouts on a Western diet. They get these basically PVCs or some kind of EADs. And so he was seeing the average of traces that didn't have that and the traces that did have that. So we're very excited. It's the first time we've actually seen what you might call a real spontaneous arrhythmia in a mouse. We have to give them a Western diet to do that, to give them that. But it happens without us doing any pacing or without us opening the mice up or anything. And so the reason we only have 3 traces in there is because we studied 10 mice of each sex and each genotype. And before we could even do EKGs on the females, 7 of them died around about 10-15 weeks. So notice the high susceptibility of the females compared to the males. So the sort of summary of this slide is that the KCNE2 knockouts get a highly diet-dependent, probably lethal arrhythmia. So for the arrhythmia, at the very least, the arrhythmia is a marker of an underlying condition which is much more marked. It doesn't happen in the wild types and doesn't really happen in the males. So almost all of them die very quickly, and then the 3 that were left in this group that we could actually study, and we'll see, by the way, QT prolongation, obviously the knockouts. The 3 that were left all had this, and about 4% of the beats have this EAD or PVC type of thing, which we've never seen before in any other mouse model we studied. And you need to be KCNE2 knockout, female, and Western diet. So it's highly dependent on those things. So if you don't worry about the sex or the age of these mice, you're not gonna see anything. So I kind of make this joke about this is the simple model I put together of what's happening in KCNE2 knockouts. But the idea is that I think certainly in the mice, and one might imagine in the humans as well, that the arrhythmia syndromes, even monogenic arrhythmia syndromes, can be highly complex, and especially in cases such as the KCNQ1 or KCNE2, where these are expressed in so many different tissues in the body. They're all over. They're in the brain. They're in epithelia. The thyroid, adrenal, not adrenal glands, sorry, pancreas, stomach. All these systems are interacting, and I think while it gets really complicated to try and think about all this stuff, I think as you saw from the few papers and abstracts that kind of interspersed, it's just starting to emerge in human populations. It is going on as well. It's not just something that happens in mice. You can see anemia in patients with KCNQ1 mutations. You can see there's a family from I think Skinner's group in New Zealand. All the people with the strongest arrhythmia also have gastric cancer and have super high levels of gastrin. So you can use gastrin in the blood as a marker for how bad the long QT prolongation is, basically. So I think in the mice we have a nice way to study it, and I think some of it at least is transferable to humans. I'm going to skip through all this because it's way too much. Okay, so conclusions. KV channels are macromolecular complexes, poor forming and regulatory subunits. We think that monogenic channelopathies can incorporate both cardiac and non-cardiac components. And by trying to use a combination of mouse genetics, unbiased, omics analysis, functional assays, we can at least get a hint about the potential complexity. And I think along with people studying, once we're all talking about the precision medicine era, where we will get much more data about patients. This type of stuff, I think, will start to emerge. When you have whole data clouds on patients, you'll find they'll have subclinical hypothyroidism, or they'll have slightly high gastrin levels, and both of those might be caused by a Casein I mutant. Both might be contributing to the arrhythmia, and they might give therapeutic windows to actually treat the arrhythmia as well. Obviously, the people are never going to have as drastic phenotypes as the mice, but I think it will be part of the big picture for what's happening. And I just want to acknowledge all the people that helped with my lab at Irvine, past and current collaborators at Cornell and at Charité, at Harvard, Jerry Berry. I didn't get to talk about stuff with him today. And then Nancy, who's the person who cloned the sodium iodide symbolika, so we worked with her when we're looking at that stuff. John Geibel did the pH measurements in the stomach. And it's my former mentor, Steve Goldstein, who gave us, along with Dan Levy, gave us the Casein IV knockout, and then Nicole Schmidt, who we're working with the Casein V knockout, Thomas Jeps, who's this fantastic postdoc who's ready to get his own lab, I think, and he did the myography studies in our lab in about 3 weeks, got some beautiful data. All right, thanks, sorry I overran a bit, thanks for listening.
Thank you, Jeff, for this vivid reminder that the system is complicated and highly interactive. And by studying ion channels in isolation, we only study parts of the building blocks of the system. So, any questions or comments to Jeff?
I want to know what's in the Western diet so I can avoid it. What do you eat every day?
Yeah, so very, the Western diet we use is a very sort of cheesy one. So it's got the kind of it smells
Sounds
really
good.
good. It smells really good actually. Yeah. It's basically got the animal fats in it, and sugar. It's a combination, right? Fat and sugar. So
Would you speculate that the problems that you see in the heart in the Casein II are problems coming from diabetes, because you don't see in the regular ones and not expression? It's not expression in any case in the heart. So
Casein II.
do you speculate that
E2 is
that
expressed
is
in the
because
heart.
of
Yeah.
the diabetes that you get the phenotype in the heart?
I think
Because
it's
you don't see when you have a normal chow diet, do you?
so we see. So what we do, we see QT prolongation without the Western diet when the mice get older. So we don't see it around 4 months. We see it around 7 or 8 months. But even on a normal diet, they're starting to become glucose intolerant at that age. So I think it's definitely a component. And they're also hyperlipidemic by that age, even on a controlled diet. So I absolutely think it's part of the picture. It's just you can speed everything up by giving them a Western diet. So.
Yeah, I lost track of how of some simple linear connection between losing E2 and having high fat and getting ventricular spontaneous beats. So where's that coming from?
So
What's it give us it in one straight line?
Yeah, that's a good question. So we think it's some kind of ischemic thing where you get some kind of ischemic damage.
So do you get fat deposits more in the E2 knockouts? Was that the ones you showed us where the undergraduate did it? So is it something to do with the coronary?
As the coronary artery, you get atherosclerosis.
And is that leading to the ventricular arrhythmia?
So those PVCs, I think, can be a marker of some ischemic damage. We haven't looked. We don't. First of all, we don't know. We published this short paper because this is only the third knockout to cause coronary artery disease in mice, basically. So we wanted to get it out there and then follow up. But we don't know whether they're really being killed by an arrhythmia. We just know that all the ones in that group, the ones that were alive long enough for us to study, have the PVC. So we don't know. We speculate it might be ischemic, some kind of ischemic damage occurring very early. But it could also be lipid composition in the membranes of the ventricular myocytes could change, because they're already probably, in the Western diet, they're already have hypertriglyceridemia and hyperlipidemia.
Do you get PVCs in the LDL model of whatever it is we're talking about, the atherosclerosis in the mouse?
In the mouse, when you...
In other words, the other atherosclerotic mouse models, do they have PVCs?
No, but I don't think so. Maybe later on, but you remember we need QT prolongation as well to see it. So I think you
Okay,
need
so you definitely have to have
we think you have the combination of QT prolongation and then whatever else the rest of the phenotype is doing. But, you know, the interesting thing is just that we have data now from a cardiac-specific knockout, Casein II, and they have a worse phenotype. So
with
I shouldn't really
PVCs?
be telling you that. No, but they have a worse phenotype. Not PVC. They just die from heart failure, from basically dilated cardiomyopathy at the age of 15 weeks. So something about one of these plethora of different things that happens outside the heart is actually protective enough to keep the mice alive despite everything that's happening. But when you do a cardiac-specific knockout and that doesn't happen, they actually die earlier. And it's a dilated cardiomyopathy. And
Is that a constitutive cardiac knockout or an induced cardiac knockout?
It's constitutive.
From
Yeah.
germline?
Yeah.
Have you looked at the histology to follow that up? Because, you know, you can get PVCs, but you just have some structural, you know, infiltration.
So we haven't done it in enough mice yet, but we've seen infarcts before in the Casein II knockouts. Yeah, we've seen infarcts, not in that group of young Western diet mice, but in older Casein IIs, we've seen infarcts occasionally, which we never see. We just didn't have enough to publish. It's just, you know, just not enough. We haven't looked at enough mice properly because you have to do an amazing amount of mice to do that properly, but we haven't had time to look at that yet. But we do see infarcts. So
Like fibrosis?
So when we get, we've done that. So when we get to 12, 15 months old, the mice get fibrotic hearts. And in the pups, the pups from knockout dams get fibrotic very early, but we think that's because of the hypothyroidism, potentially. They get multiple problems. It's very complicated if you look at that population. But when the mice get older, they definitely get fibrotic. I mean, they also get fibrotic livers as well. We think that's probably, again, even that's complicated. It's probably due to right heart failure and the fatty liver developing into NASH and you know, fibrosis, cirrhosis. Yes.
Related question. You showed a lot of your data for the multisystem failure in the knockouts versus the wild types, but you mentioned early on discussing the stomach that the current defect is present in the hets, even though they don't have
Yes.
the dysregulated morphology.
Yes.
How much of the multisystem failure translates to the hets?
Yeah, that's a really good question. We haven't looked at that. We have not looked at that. I'm just trying to think what we've done in heads. Yeah, we've barely done anything in the heads because we wanted to use the heads to answer that one question about whether it was the morphology of the parietal cells that was causing it or whether it was a primary defect. So we looked at them, but we don't look at heads that often. We've looked at a lot of differences in terms of the maternal genotype and the paternal genotype because of hypothyroidism, and we figured out that the pups are fine. Even knockout pups develop normally if the mothers are wild type or heterozygous. So that's really important. The maternal genotype is really important. And one of the reasons I figured out what was happening there was because I noticed that knockouts usually are smaller than the pups, but the knockout dams were getting enormous when we were doing the micropesting. What's going on here? Then I realized they're just not ejecting milk. So they're getting so big because they can't get rid of milk, so the pups are getting starved. And the small amount of milk they do get is very low in iodine or thyroid hormone or both. So when we injected them with oxytocin, so hypothyroidism causes milk ejection defect. Even if we inject them with oxytocin, they release the milk, and they were fine. So my answer to the heterozygous question, we just haven't done that much of it. It's a really good point because we might start to see a bit of a mixture of more subtle effects, but at the same time, you know, when you're trying to figure out mechanism, it's easier to just have it gone. Right?
So you have it's very nice that you show that Casein II has all these effects, but from what you said, I seems that the Casein II only interact with Casein Q1. That is the only partner. You didn't find other partners.
Oh yeah, so yeah, I mean, I wanted to give kind of an overview today, but I probably tried to talk about too much. So basically, in epithelial systems such as the thyroid, the choroid plexus, which I didn't talk about today, and the stomach, Q1 seems to be a major part of Casein II. Maybe in the pancreas as well, but we haven't looked at that. We're doing it, but we haven't come to any conclusions yet. In the heart, in the mouse heart, Casein II interacts with Kv4.2 and Kv1.5. So it upregulates both of those currents. So it upregulates ITO-F and IK slow, the 4-aminopyridine sensitive portion of IK slow. So when I didn't show that data today, but in younger mice without all of the accumulation of all this other stuff, you see a straight ventricular defect, a reduction in repolarization reserve because you reduce the density of IK slow and ITOF.
So the what the consequence of like the mice die in a young age. The consequence, is there anything that's contributed by other partners of the like Kv4 and 1.5?
Without the QT prolongation, I don't think there would be. I think if that arrhythmia is really lethal in those mice, they need both the QT prolongation and the ischemic or whatever's happening. I think it's a combination of both. That would be my best guess. But because it's so difficult to get ventricular arrhythmias in mice like this anyway. We do when you do a coronary artery ligation, you can cause without ventricular fibrillation in the knockouts. So I didn't show you that data, but so if you push the ischemia further, the knockouts are highly susceptible. They'll get ventricular fibrillation, which is quite difficult to induce in mice because they have small electrical reasons. So the QT prolongation is a hugely important component of the at least the lethal arrhythmias anyway. Coronary artery disease, I think it's related to dysfunction of Casein Q1, Casein II channels in multiple different tissues. That'll be my best guess. Yes, Chief.
So when you say that the KCNE interacts with, are you talking about the functional assay output or real biochemical evidence that they actually
Really, yeah, biochemical evidence. Yeah.
So they can be immunoprecipitated
Yes.
from intact tissue?
Yeah, in the case of Casein II, we actually got in the ventricle tissue, we got coIPs with Kv1.5 and Kv4.2. And those are the only two currents that we saw changed. Yeah. They interact. Now, as far as I remember, so Casein II seemed to be important for getting Kv1.5 to the intercalated discs. So when we knocked out Casein II, we saw a big hole where normally we'd see Kv1.5 staying in intercalated discs. I can't remember what happened in CHO cells, whether we saw that effect or not, so it might be highly myocyte specific. Whereas in Kv4.2, you can recapitulate that in multiple cell types. Casein II always upregulates it, it slows its inactivation a little bit. But Kv1.5 it seemed to be mostly a trafficking issue. It basically, and it was very ventricle specific. In the Atria, which I don't know if we published yet, we didn't see any effect for Casein II and Kv1.5 trafficking. So in the anterior ventricle, it's different. A lot of it might require certain other elements of the secretory pathway. So, in the stomachs, for instance, we knocked out Casein II. Casein Q1 goes to the wrong side of the cell. It goes to the basolateral membrane. So Casein II is important for Casein Q1 getting to the apical membrane and doing its job there. So we knock out Casein II. Q1 goes to the wrong side of the cell, but it's because of upregulation of Casein III. So when we found that Casein III is upregulated, we knock out Casein II as well. When we knock out Casein III as well, you have a double knockout. Q1 goes back to the apical side. So it doesn't require Casein II to take it to the apical side. Just Casein II stops Casein III from hijacking it and taking it there. Now normally Casein III is not that highly expressed in driver cells, but for some reason when you upregulate when you knock out Casein II, Casein III this remodeling goes on, and it kind of snatches Q1 away and takes it to the wrong side. Yeah, so there's a lot of trafficking effects of KCNEs as well, and they're different depending on the cell type. Because in colon, Q1's at the basolateral side, the opposite in the stomach, because KCNE E3 because it's partner in the colon. Yes.
Okay. Oh,
Go ahead.
there's more.
Yep.
No. It's your turn.
Oh, okay. The Casein III reminds me of a paper I saw a long time ago, which was cardiomyopathy. Hamsters live longer if they're raised in constant light.
In constant.
Constant light, 24 hours a day, 7 days a week, constant light.
Very interesting. Well, that's Tracy's.
They had bigger hearts. They lived longer.
Why are you talking? So there was a paper we were discussing. There was a paper in Nature a year or two ago showing circadian rhythms, and I think it was K-chip was regulating ITO.
Yeah.
So these ion channels are, they're regulated by hormones. They're regulated by circadian as well, because we need higher and lower potassium currents at different times of day. Certain arrhythmias are highly dependent on the time of day, as we were discussing earlier. Brugada patients tend to die in their sleep, and hERG mutations tend to kill you when the alarm clock, right, in response to the alarm clock. So we try and do our studies at the same time of day, every day, because we're aware of these oscillations. Yeah, it makes a lot of sense. It wouldn't surprise me at all. Someone needs to do the microarray or transcriptome analysis on the hearts in constant light compared to the regular light cycle, and look at I bet you everything changes.
It's in the New York Academy of Sciences,
Oh,
but
yeah.
I've looked for it, but I haven't found it again yet.
That's interesting.
All right, we still have to deal with global warming. So,
Yeah.
thank you.